Okay guys, this is lecture 10, Living in Water, which is effectively about osmoregulation, and the subtitle is Drinking Ions and Urea. So these are the learning outcomes, and what you want to concentrate on is how organisms osmoregulate in the face of being hyperosmotic or hypoosmotic to their environment. So if we look at the learning objectives, we've got to examine the osmotic potential of organisms in freshwater and seawater. So the majority of organisms are going to be hyperosmotic to freshwater and hypoosmotic to seawater. The second objective is how organisms regulate their osmotic potential, ionic composition and cellular volume. Okay, so osmosis or osmotic potential is related to, it can be related to ionic composition and will be related to cellular volume. And thirdly, we'll examine the evolution of different mechanisms to ensure osmotic, ionic and cellular volume regulation. A couple of keywords there: TMAO, which we'll talk about with reference to urea, urohaline and sterihaline. You should be familiar to both of those. Urohaline, you can live in a wide range of salinity. Sterihaline, you can only live in a specific salinity. Hypoosmotic, which means you've got a lower osmotic concentration inside you compared to outside. Hyperosmotic, you've got a higher concentration inside you of solutes than outside. And salt glands, which are obviously glands that excrete salt. Alright, these are chapters in the textbooks, and there are two references following which I recommend you have a quick look at when you get a chance. So the first reference looks at osmoregulation in marine mammals. Okay, marine mammals will be hypoosmotic to their external environment, and they also need to get water from somewhere because they live in the marine environment. So, and obviously seawater is going to be hyperosmotic to them. And the second article is a review looking at osmoregulation in elasmobranchs, which tend to be actually isoosmotic or hyperosmotic to the external environment, which is just about always the sea. Okay, and they use a specialised system of urea and TMAO in order to achieve that. Alright, so for this lecture and the next lecture we're talking about living in water. So what's the problem about living in water? We've obviously talked about respiration, but here we're talking about osmotic potential or osmoregulation. So why would living in water and your internal versus external composition be important? Well, simply, if we look at the table, we can see that seawater is iron rich and freshwater is iron poor. Seawater has lots of ions in it, and seawater is usually hyperosmotic to organisms, whereas freshwater has very few ions in it, and freshwater is normally hypoosmotic to organisms. And this means that there's an osmotic gradient between an organism in freshwater or in seawater. So in freshwater, what will happen is water will want to flood into the organism to try and reduce the hyperosmotic environment of the organism compared to the seawater, whereas in seawater, water will want to flood out of the organism to reduce the hyperosmotic environment, hyperosmotic external environment that the organism is in. So if we look at seawater specifically, okay, we used to say it had a salinity of 35 parts per thousand. We're no longer allowed to use that. We have to say it's 35 PSU, which are practical salinity units or dimensionless units. And basically what it means is we've got 35 grams of salts per litre of seawater. And when we talk about salts, the principal salts in seawater is sodium chloride, but there's also a little bit of magnesium, calcium and some sulfate. And talking about the marine environment, this marine environment is responsible for 69% of the Earth's surface, so the majority of the Earth, and it's where all organisms came from. So here we've got a table of the main salts that are found in soft lake water, river water, hard river water, saline water or seawater is what it means, and the Dead Sea. And we've already said that seawater is responsible for 69% of the Earth's surface. Fresh water is about 1% of the Earth's surface, and estuarine is about another 1% of the Earth's surface. Okay, so estuarine we define as somewhere between 0.5 to 3% salt concentration, or that's 5 PSU to 30 PSU, and fresh water has to be less than 5 PSU. And what we can see is that there's a range of different solute concentrations or ion concentrations. So soft lake water has hardly any ions in it. Harder river water is about a magnitude of order bigger. Seawater is, as we then jump 2 orders of magnitude, so we go from 10 to 1,000, and then when we look at the Dead Sea, we've jumped another order of magnitude where we're up to 10,000. Okay. if I said to you the reason the Dead Sea is called the Dead Sea because there's no living things in it apart from some very few weird bacteria, what does that tell you about the effect of osmotic potential upon organisms? So the concentrations of ions between these different environments, between freshwater and seawater, differ. So what's the problem with that? Why are we bothered about ion concentrations? So if we go back to good old Claude Bernard, who defined the internal environment, or the mu interior, he said that the cells within an organism experience their own internal environment, different to and distinct from that occupied the organism, which is the external environment. So basically, as you should have inferred already, and as I've mentioned, in freshwater organisms, their internal ion concentration is much greater than that of than the concentration of ions externally. So basically, they're hyperosmotic to the hypoosmotic environment, whereas in seawater, we have the opposite situation where higher order organisms tend to be hypoosmotic internally to the hyperosmotic environment. However, if we look at lower order organisms, we can see that lower order organisms tend to be isoosmotic to their external environment. That is, they have the same number of ions or solutes dissolved in them as the external environment does. Okay, as well as differences in the total concentrations of ions, so hyper versus hypoosmotic, what else can you see? Well, if we look here, we can see, okay, the total concentrations of the ions can be different, but also the individual concentration of a particular ion can be very different from internal versus outside. And if you think about it, this has to be obvious, because how do we know life exists? Because biological gradients exist. If there aren't gradients or differences that we can see, then everything's the same and everything's not alive. Alright, so what does all this mean? There can be different concentrations of ions, and the number of and for any particular ion there can be a different amount of it internal versus outside. Alright, so this is where we start talking about osmosis or osmoregulation. So unless an organism has a total concentration of ions or solutes, so that's things dissolved in it, equal to the number of things dissolved in the environment. So if you remember for osmosis, it doesn't matter what's dissolved so long as the total number of dissolved things are the same, okay, then you've got a problem, you're subject to an osmotic stress. So what do I mean by it doesn't matter what it is? So if I have 100 sodium ions in a cell and I have 100 proteins outside the cell, then there's no osmotic difference between the two things, they're both at 100 milliosmoles. However, if I have 100 sodium ions inside the cell and I have 50 sodium ions outside the cell, then the cell is hyperosmotic to the hypoosmotic outside environment. Okay, so if there's an imbalance, if there's a difference in the number of solutes inside versus outside, okay, then one is going to be hyperosmotic and the other is going to be hypoosmotic. Now, typically, organisms are normally around 300 to 500 milliosmoles. Right, there are some exceptions and we'll talk about that. When we look at the osmolarity of seawater, it's normally around 1000 milliosmoles. Fresh water is about 0.5 to 15 milliosmoles, and brackish water, so estuarine water, is about 15 to 850 osmoles. So, therefore, in fresh water, organisms are always going to be hyperosmotic to the hypoosmotic external environment, and in seawater, organisms are normally going to be hypoosmotic to their hyperosmotic external environment. Now, these have some important consequences. Alright, so unless an organism is isotonic or has the same concentration of number of solutes dissolved in it as outside, it's going to be subject to an osmotic stress. The result of that osmotic stress is that there's going to be an influx or efflux of water. So if the organism is hyperosmotic, water's going to try and come in. If it's hypoosmotic, water's going to try and release. Okay, this can lead to a change in the concentration of particular ions. Okay, and this loss or gain of water is going to cause a change in cell volume, so either an increase in cell size or a shrinkage. Okay, just for a bit of an aside, watch this little video before you move on to the next slide. So organisms living in fresh water or in the sea are going to be faced with either osmotic regulation or conformity. Okay, so that means they can choose to be isotonic with that environment or maintain their hyperosmotic or hypoosmotic status. Okay, then they've also got ionic regulation and conformity, and that means that do they ensure that they have the same composition internally as externally, or maintain a different composition? And okay, for some ions it's going to be the same, but for other ions it's going to have to be different, because otherwise they're going to be the same inside as outside. And then finally they've got to deal with volume regulation and conformity. So they either maintain cell size, or as the environment changes, do they allow their cells to swell up and shrink. So what we've got here is we've got three different species. On the top two plots, we've got an isoosmotic line. So what happens to the blood osmotic pressure as we change the ambient osmotic pressure, and you can see a regulator keeps its blood osmotic pressure constant irrespective of what's going on outside, and a conformer simply changes according to what's going on outside, so just as we see with temperature regulators and temperature conformers. And on the bottom plot we've got three species. We've got a green crab, we've got a shrimp, and we've got a mussel. Now the green crab's the blue line, the mussel's the red line, and the shrimp's the green line. Okay, and we can see quite clearly the mussel's an osmoconformer. The green crab seems to regulate to start off with, but as the osmotic pressure rises, it begins to just become an osmoconformer, whereas the shrimp pretty much is an osmoregulator through its entire range. And if we look at some definitions, and you've seen some of these before, if an organism is isoosmotic, its osmotic potential of its internal environment is the same as the external environment. Hyperosmotic, it means that your internal environment is, the osmotic potential of it is greater than the external environment. Hyperosmotic, osmotic potential inside is less than that outside. An osmoconformer is an animal whose osmotic potential will match that of its environment. An osmoregulator is an organism whose osmotic potential will remain constant despite changes in the external environment. Urohaline is a species of organism that tolerates a wide range of osmotic potentials. Stenohaline, it can only live within a very specific or narrow range of osmotic potentials. So if we go back to our crab, mussel and shrimp species, I would say the shrimp was definitely a regulator, an osmoregulator. The mussel was definitely osmoconformer, and the green crab, depending on the osmolarity, is somewhere in between. If I was to talk about which species is urohaline or stenohaline, I'd say the shrimp and the mussel are stenohaline, because it doesn't really matter. But because we've got a change in the osmotic potential of the crab as we change the external environment, I would say that the green crab is probably stenohaline, because it's attempting to regulate, which means it can, but then it sort of gives up as the osmotic potential outside rises too much. Okay, and remember, organisms will live in very variable environments, particularly in estuaries. So here we have, this is estuarine shore crab from Cheapskate Bay in the States, and we can see that the salinity in the bay can range from 5 up to 30 parts per thousand, or sorry, 5 to 30 PSU. Okay, and if we look at the osmotic potential of the crab, we can see actually across the normal range of experiences within the estuary, it's effectively an osmoregulator. So unless you're a conformer, you must regulate, and remember that to some extent, well, to an extent, all organisms are going to have to regulate for some ions, otherwise they're going to be the same inside as outside, which means they're not alive, they are the external environment. So all organisms at some level have to exhibit regulation. So if you are going to regulate, how are you going to achieve this? And there's three different ways I can think about organisms can achieve regulation. So before you move on to the next slide, I want you to think about the three different ways that you could regulate ion concentrations in an aquatic organism. How can you ensure osmotic regulation to some extent? Okay, why did I mention terrestrial organisms? Okay, well, one thing you can do, okay, if you live in a hypoosmotic environment, is wheelots. Okay, water's going to try and flow into you, but if you keep excreting that water, okay, then you should remain hyperosmotic. Okay, obviously if you live in a hyperosmotic environment and you're hypoosmotic, you're not going to wheelots. Okay, so regulating the amount of urine you produce can help regulate your ionic composition and your osmotic composition. The second thing we could do to affect our osmotic potential is through active transport, so we can either actively excrete ions, okay, into the external environment, or actively take them up in order to achieve both an osmotic and an ionic balance. Now the third and perhaps the easiest thing to do is basically we have a carapace or a shell that's impermeable to water. So that's great, it doesn't matter what we are inside compared to outside, nothing from outside can get in and nothing from inside can get out. But we're living in water and there's a problem with that. Okay, in a terrestrial system that's not an issue, but in water that is a problem, in that because we live in water and because water is so crap at holding oxygen, we need to have gills. So that means that irrespective of having like a big impermeable shell, part of our body is always going to be exposed to the external environment, so we're always going to have some sort of regulation there. So what's another way we could look at regulating our osmotic potential without having elaborate pumping or impermeable carapaces and all the rest, and without affecting our cell volume and things like that? So we are effectively isotonic or isoosmotic with our external environment, but we've still maintained a different ionic composition, etc. What's the easiest way to prevent water influx or efflux without actually actively excreting water or coming up with some elaborate mechanism? Okay, well the simple answer is we produce what we do is we produce a solute, okay, which we excrete into our cells, so we have the same concentration of solutes, dissolve things in our cells as we do outside, so therefore we're isoosmotic with the external environment, so no water is going to come in or out. Yes, there's still going to be a difference in the type of ions, so we're still going to have ionic gradients, but we're not going to have an osmotic gradient. And so this is what some organisms actually do. So what they will do is, in response to either living in a hyperosmotic environment or to a changing osmotic environment, they will actively excrete molecules into their intracellular fluid or solutes into their intracellular fluid, so that the solute concentration intracellularly balances that extracellularly. Typically, most organisms that do this will excrete amino acids that have little metabolic impact, so we're talking about glycine, serine, beta-alanine. So why are they excreting amino acids, which are quite complex molecules? Why don't they just put more potassiums or sodiums into the extracellular into the intracellular fluid? Well, if you think about it, the most abundant ions we normally talk about are sodium and potassium. Do we really want to excrete sodium and potassium intracellularly? No, because that's going to change our membrane potential, which is going to affect how reactions occur, how we respond, cell communication, all the same. And if we started secreting active amino acids, then this is going to change the rate of reactions, the same as if we excrete ions, which are important. So by excreting non-active waste metabolites, okay, what happens is we increase the solute concentration, so we balance out, but we're not affecting the reaction processes that are going on within the cell. Okay, just as an aside and just as a note, remember when we talk about higher order organisms, okay, set regulation occurs at a plasma, at a tissue level rather than a cellular level, and occurs across at many levels because we have many different internal and external environments in vertebrates, for example. So here we've got a series of marine invertebrates with one exception of a hagfish at the bottom. And what I want you to do is add up the biggest ions across there and see whether or not it's different to 1,000 milliosmoles, which is the concentration, which is the osmotic potential of seawater. And if we add them up, we can see they're all roughly around 1,000 milliosmoles, okay, which means that most marine invertebrates are osmotic conformers. That is, their intracellular osmotic potential is equal to the extracellular osmotic potential. So their osmotic potentials are the same, but are their ionic compositions the same? Well, your obvious answer is, duh, no, Seb, if they were the same inside as outside, they would be the outside environment, they wouldn't be alive. So, yeah, here we've got a plot of percentage of body fluid dialyzed against seawater. So if the number is less than 100%, that means that there's more of the ions in seawater than there is outside. And if the ions number is greater than 100%, then it means there's more ions intracellularly than there is extracellularly. And we can quite clearly see here that although these organisms are isoosmotic with seawater, as we would expect, some of the ions are the same, but some of the ions have very different concentrations inside versus outside. And what is this telling you, and how are they? It's telling you they're alive, because they must be, because they're generating a gradient. How are they doing it? Well, how would you normally maintain an ion concentration? There must be some form of active transport going on. So if we look at organisms that occupy estuarine or brackish environments, okay, so they can go from living in a hyperosmotic environment to living in a hypoosmotic environment. And we can see we've got some species such as the European shore crab, which can osmoregulate down to 22 parts per thousand or 22 PSU, and then just basically osmoconforms and dies. But then we have other species such as brine shrimps, grace crabs, fiddler crabs, okay, which don't really care how the osmotic potential of the external environment changes, they manage to keep osmoregulating. So these will be the true successors, or the true successful species in an estuarine environment. Now, just as we can acclimate organisms to different temperatures, we can also acclimate organisms to different salinities. So we would expect to see an acute response if we suddenly change the salinity, but if we acclimate our organisms for a long enough time, okay, what happens is they respond by developing a heartbeat, or here we've got ciliary activity, at the same rate as organisms normally subject to the environment. So what we've done here, we've taken Baltic Sea mussels, which live at low salinities, and we've taken North Sea mussels, which live at high salinities, swapped them around. We can see that ciliary activity is very different to start with, okay, but then over time, as the Baltic Sea mussels mimic that when moved into the North Sea, mimic that of North Sea mussels, whereas the North Sea mussels over time mimic, which come from a high salt environment going to a low salinity environment, mimic the ciliary activity of Baltic Sea mussels. So acclimation is not just restricted to temperature. So freshwater organisms are always going to be hyperosmotic to the hypoosmotic environment. So this means that they always have to regulate, otherwise they're going to burst from all the water that's flooding into them, trying to make them isoosmotic with their external environment. So what's the easiest way to deal with an influx of water into you? Simply wee it out. So urinate lots of water back out. The only other problem with that is, although you might be very efficient and have the pinnacle of excretory evolution, you're not going to absorb all of the ions, so some of that urine is going to contain ions that you don't want to lose, that you are going to lose, so you use a combination of urinating lots and also actively transporting back in from the external environment essential ions. So if we look at this slide here, what we can see is that some marine fish appear, or some so obviously when we talk about marine fish, we're talking about vertebrates. Some fish are isoosmotic with seawater and some are hypoosmotic. So all the elasmobranchs here, apart from a silly freshwater elasmobranch, okay, are isoosmotic to seawater, whereas all of the teleosts are hypoosmotic to seawater. Well, surely they all evolved from the same ancestors, so why are elasmobranchs isoosmotic and why are bony fish, teleost fish, hypoosmotic? So if we look to the previous slide, we could see that elasmobranchs, hagfish and coelacanths are also isoosmotic to seawater, whereas bony fish weren't. They were hypoosmotic to seawater. But if we look at their evolution relationships, we can quite clearly see that bony fish, so these would be sturgeon, trout, bass, carp, etc., are more related to coelacanths, okay, than they are to teleost fish, whereas teleost fish tend to be more related to hagfish. But coelacanths are isoosmotic to seawater, as are hagfish. Why are bony fish different to teleosts? And why does there seem to be confusion in this phylogenetic tree? All right. Well, before we look at that, how do elasmobranchs manage to be isoosmotic with seawater? Well, just like simple invertebrates, why don't you take a waste metabolite so it doesn't cost you any energy to produce it, and rather than excrete it to the external environment, excrete it into your intracellular fluid. So long as it's a solute and so long as it's non-reactive, it's going to ensure that your internal solute concentration matches that of the external environment, therefore you're isotonic with the external environment. And this is exactly what we see elasmobranchs and coelacanths doing. Instead of excreting all of the urea to the external environment, they retain some inside, such that their internal concentration is solute concentration is equal to or greater than the external solute concentration. So living in seawater as a shark, I can be isotonic or even hyperosmotic to seawater, meaning there's no need to drink or do anything else like that. Water's going to come into me. Brilliant. But there's a problem with that. we wouldn't want to store ammonia inside us because ammonia is toxic, and it's the same with urea. Now maybe if we converted urea to guanine it would be okay, but then I would say, well hang on, surely urea or guanine and uric acid don't need water, which is why we see desert organisms excreting it. So if we had the same concentration of urea in us as elasmobranchs do, so it's 100 times greater than our normal urea concentration, we'd be dead. So how do elasmobranchs manage to have such a high internal urea concentration and not die? And the simple answer is they excrete a buffering compound, or enzyme if you like, called TMAO, or trimethylamine oxide. And what that does is it basically negates the denaturing effect of urea, or stops urea from denaturing their internal proteins, making urea a safe solute to excrete internally. Okay, so that explains why elasmobranchs and coelacanths managed how they achieve isoosmotic potential for their external environment. But why are hagfish isoosmotic for their external environment, and why are teleost fish hyperosmotic? Why aren't they the same? So with bony fish, what we used to believe, or what the story was that we believed that we have the elasmobranchs. Somewhere along the line of the ancestors of the elasmobranch decided to go and live in fresh water. So if you're going to live in fresh water, you want to reduce your solute concentration internally, otherwise it's going to be too much stress. So what we said was that bony fish evolved in a freshwater environment, and then the bony fish we now see in the marine environment are derived from freshwater ancestors. So teleost fish evolved in the marine environment and always stayed in the marine environment, whereas bony fish evolved in a freshwater environment, and then some of them moved from that freshwater environment back into the sea. It's a nice story, but is that really so? Go on then, you can play the Homer video for a break. Okay, and just talking about teleost fish living in freshwater and a marine environment. Okay, where in their freshwater they're obviously not going to drink and excrete, they're going to urinate a lot and actively transport salts back in. Whereas in seawater, what they're going to do is they're going to drink lots of seawater, retain the water, and excrete lots of salt, because what they want to try to do is they're trying to keep the water's trying to move out of them. They're trying to get as much water back into them as much as possible. Urination will be low. Drink lots of water, excrete the excess salt. So, did bony fish evolve in a freshwater environment and then move back into the sea? Well, remember I looked at that phylogenetic tree and we said that hagfish were closer to elasmobranchs, whereas coelacanths were closer to bony fish. Well, hagfish, okay, are isoosmotic with their environment, but they don't actually use urea. So this suggests to us, and we now have changed our mind, rather than believing that bony fish evolved in freshwater and then moved back to the sea, we believe that bony fish evolved in the sea, and then for some reason there was a selection pressure selecting, there was some benefit to being hypoosmotic into your external environment rather than being hyperosmotic, and this hypoosmotic, or the lower osmotic potential of bony fish, allowed them to move from the marine environment in order to colonise freshwater environments. So remember, if we look back very early on the slide, there was only one elasmobranch that lived in freshwater. Okay, if we look in freshwater now, right, there's very many different species of bony fish that have inhabited freshwater. So this reduction in internal osmotic potential in the marine environment has allowed freshwater fish to colonise freshwater environments, but there also must be some other advantage associated with having a lower osmotic potential in a marine environment compared to being isoosmotic. And we'll talk about that in another lecture. So there's only one amphibian that lives in the marine environment, and that's Rana carnivora, which we've come across before, which is the crab-eating frog, who lives in seawater because it's hunting crabs, obviously. So how does this crab, which has an internal concentration of around 300 milliosmoles, manage to survive and live in seawater? And the answer is, just like good old teleost fish, just like good old elasmobranch fish, it uses urea to make itself isoosmotic with its external environment. Alright, I'm a marine reptile, a marine bird, I'm an albatross or something like that, or I'm a mammal, okay, and that means I live at sea for the majority of my life. So where am I going to get my fresh water from? I need fresh water. I am going to lose this water out of my body. Metabolic water is not going to replace it. So I might drink seawater, but as a human, if I drink seawater, I'm going to die. Remember William Taylor Coleridge's poem, water, water everywhere and not a drop to drink. Water, water everywhere and all the balls to drink. So you can drink seawater, but for us it will kill us. But for marine reptiles, mammals and birds, what they can do is they can drink seawater, and what they then do is they'll actively excrete the salts contained in seawater, so leaving them with fresh water inside their system, and then they'll excrete the salts out through salt glands. So if you, any of you know seabirds, if you know anything like a fulmar, they've got two little tubes at the top of their nose, and what they can actually do is if they feel threatened, they can snort out, these are their salt glands, and they can snort out the salty fluid into your eyes or into your face to try and scare you away. And if you watch the iguana video on the next slide, you can see the iguana after a while snorting out salt from its salt glands. And that's it.